Hello everyone, my name is Bao-Wen Gai. I'm a PhD student from the 6th Affiliated Hospital of Sun Yat-sen University in China. My topic is translating molecular subtypes into clinically applicable radiogenomic biomarkers for survival prediction of colorectal cancer. Colorectal cancer is a common malignancy of the gastrointestinal system, with the third highest incidence and the third highest mortality rate in the world. CRC is a highly tumor heterogeneous cancer, and the existing TNM staging doesn't take this into account enough to fully meet clinical needs. To allow better classification of CRC based on its tumor heterogeneity, the International Consortium of CRC Subtypes identified 4 consensus molecular subtypes, the CMS. However, it still has limitations as a molecular subtype and is not easy to achieve clinically because it requires high throughput sequencing as a disease. Radiogenomics has potential for molecular typing, disease diagnosis, and treatment prediction, and is more clinically useful due to its lower cost. Therefore, we want to establish a general framework for translating molecular subtypes into low-cost radiogenomic biomarkers, providing more accurate prognosis prediction and treatment guidance. A total of 2,953 CRC patients from 8 cohorts were enrolled in this retrospective study. In addition to the public cohorts shown in the figure, we also use a cohort from the SGC-ARGO project and an in-house radiogenomic cohort from the 6th Affiliated Hospital of Sun Yat-sen University. Let me introduce the COCC cohort here. COCC refers to the Colorectal Cancer Clinical Genome Study in China. It's a colorectal subproject of the SGC-ARGO. This project is led by the 6th Affiliated Hospital of Sun Yat-sen University and BGI Shenzhen. The project plans to sequence samples from 2,000 Chinese colorectal cancer patients, and currently RNA-seq data from 587 samples were available. Next, I will present an overview of our study design. This study is conducted in 3 major steps. First, drawing on previous work in the laboratory, we transformed high-throughput gene expression data from tumor samples to functional spectra by gene set enrichment analysis. Using the obtained functional spectra as input, subtypes of CMS were used as classification results, and a classifier was trained using deep learning. And then the last hidden layer was extracted as CMS-associated deep features. Second, in order to develop a clinically available assay based on deep features, we initially calculate the top 10 genes or radiogenomic features associated with each deep feature. We then selected the top 3 variable genes or radiogenomic features for each deep feature to obtain CMS-associated signatures. The CMS-associated signature were used to predict the prognosis of CRC patients. Last, we employed survival analysis to illustrate the prognostic disparities between patients in the high and low-risk groups. Additionally, functional analysis was conducted to provide a biological rationale for CMS-associated signatures and offer guidance for immunotherapy. I will go on to show what we found. As seen in the figure on the right, our CMS-associated signature can be a good predictor of a patient's prognosis. Better results were obtained on the training cohorts as well as on the 3 validation cohorts, and the differential genes in the high-risk group were all enriched in the extracellular pathway, which is consistent with the biological characteristics of CMS4. and it's CMS4 that has a relatively poor prognosis. This confirms that our approach can rely on biology of molecular subtypes to better distinguish patients' prognosis. In addition to this, we also found that patients in the low-risk group were more likely to benefit from immunotherapy based on TIDE, IPS, Miracle, and TMB scores, which may make a contribution to individualized and precise treatment. Further, we developed a radiogenomic signature and found that it still predicts patients' prognosis well. Moreover, the radiogenomic signature is highly correlated with the relevant biological pathways of CMS. Now, let's summarize this study. In conclusion, we established a general framework for translating molecular subtypes into low-cost radiogenomic biomarkers, providing more accurate prognosis prediction and treatment guidance. And our findings have enabled a bridge between molecular subtypes and clinical applications. Moreover, it not only provides more help to clinicians in making treatment decisions, but also greatly reduces the cost of medical treatment for patients. That's all I want to share. Thanks for listening.